Hello everyone, welcome to my YouTube channel. So here today we are going to be looking at the macromolecules, a section of the macromolecules which is our proteins, which is a continuation. So macro, we said that in the previous video for carbohydrates. Macro means big, so these are just big molecules. So proteins are made up of amino acids. So made up of amino acids. Now, so proteins are made up of amino acids, which are basically amino acids are the monomers. So monomers are just subunits that are used to make polymers. Polymers are the large molecules. So macromolecules are basically polymers, which are formed as a result of the combination of many monomers. So now, when we look at our monomer, which is our amino acid, so the basic structure of an amino acid is that an amino acid will look like this, OH, like that. Then we are going to have another, so this is what we have. We might have hydrogen there, others might put R there. Then we are going to have our R like that. Then we are going to have this. So now the amino acids are formed as a result of having a carboxyl group. So this one is what we call the carboxyl group. So you can just add the group. So this is the carboxyl group. We have the functional group, which is the R group. So any amino acid will only be different by R. R could be anything. It could be glucose in the case of glycine. It could be other things such as CH3 in the case of alanine and all that. So we have group there. Now, when we look at this, we have then this one is what we call our amino group. Not amino acid, but amino group like that. So that is our amino group. Amino group like that. So this is basically the general structure of an amino acid. So now let's draw it in other ways. We can have our C like that, C. Then our carboxyl is drawn like this. Then we have our OH like that, which is the same as that. Then here we are going to have it is bonded with a nitrogen like that. So we have our hydrogen. We have our hydrogen. In fact, to have more precise, we can write it is still NH2 like that, but hydrogen is the one that is bonding to three things, so we can write like that. So this is the structure of an amino acid. So how do they join? So the joining of amino acids looks like this. So here we had our H there, we have our R group there. So when amino acids combine, we are going to have, when the amino acids combine, we are going to have this type of a structure. So remember, these combine in such a way that we are going to have this type of a structure like this. So we have, it will be a dehydration reaction in which water has to be released. So as you can see, this is our water, so this water will be released. So we are going to have our H2O gone like that. So we are going to, when this happens, we are going to have a double bond O there. We have our C, we have our H, we have our R, we have our N, we have our H, we have our H. So we can even say R1 to show that it is a different one. Sometimes they can combine with themselves. Then we are going to have a bond. So as you can see, water is taken. So this new bond, this one will bond to nitrogen. Then we have our hydrogen there. Then we are going to have everything is the same now. We have our R2 there. We have our H. We have our C. We have double bond O. We have our OH like that. So this is how the bonds take place. So now these are held together by what we call peptide bonds. Number one, we might talk about nonpolar. So nonpolar, these have got the side chains. These have got the side chains. So firstly, when we look at nonpolar, they do not dissolve in water. That's the meaning of nonpolar. So how we draw our amino acids, we have nonpolar side chains where we have mostly, we are going to have, if we have, let's say, H for the R, where we could just have CH3, we could have different things. If you want how these structures are drawn, you can just comment and it shall be sent in the description below, or rather in the comment section. So now, if you look at the nonpolar, let's draw a simple structure. Let's say we have C, we have C like that, double bond O, we have OH like that. Then here we have our H like that. Then here we had our R. Here we have our N, we have our H, we have our H. Now for R, here, if we have on R, we have H. So this is an example of a nonpolar, which we can say this is our glycine. So we have our glycine like that. Then the other thing that we might have is, if we change this with CH3, if it looks like this, then we have our alanine. So other structures have been drawn in a separate video. So we have our glycine, we have our alanine, we have our valine. So this one is alanine specifically, our CH3 there. Then we have our valine like that. We might have our leucine. We might have our leucine. We might have our isoleucine like that. We might have our methionine. So all these are nonpolar. Other structures which are for, we might have our polar, which when we say polar, we mean to say they are hydrophilic, meaning to say they are water loving. They are uncharged, but they are able to dissolve. So they are able to dissolve in water. So these are able to dissolve in water. Now, when we look at these, we might have examples such as our serine or serine. We might have our 309. We might have our 309. We might have our asparagine, glutamine, among others. So now, the thing that is peculiar about these is that they mostly have hydroxides on their side chain. So they mostly have hydroxides on their side chain, just not to say they dissolve in water. Just to take one example, let's look at serine, for example. We know that it's N, so we have C, we have H, we have OH like that. Then for serine, here we are going to have CH2 like that. Then here we are going to have our OH like that. So this is the structure of serine. So most of them have got they have got an OH on their side chain like that. They have an OH on their side chain. So this makes them soluble in water. They are able to interact with water like that. We might also have what we call polar, which is charged, polar charged. That one was polar uncharged, or rather ionizable amino acid. So these carry a charge. The examples that we might have, either they might be acidic amino acids. So this one might be acidic amino acid. We might also have basic amino acid. First, our acidic amino acid, an example that we might have is aspartic acid. So we might have aspartic acid. Then for basic, so these are charged. As you can see, an acid is charged. Then if we have a basic amino acid, so I can just write AA, amino acid. Basic amino acid, we might have glutamic acid. Oh, sorry, this one is acidic. Then if we have our basic amino acid, I can just say amino acid, we might have these examples. A number of them. We might have what we call lysine, we might have what we call arginine, we might have what we call histidine. So histidine, this one is also an example of an amino acid that has got an aromatic ring. So now that's not the only thing that we have. Others, though they might be polar, non-polar, but others have got the special functional group. So special functional group. So now the special functional group So the special functional group that we might have is, number one, we might talk about glycine. So if you recall how I drew glycine, I drew C, then where there is R right here, we just put H, if you recall. So this is a special amino acid. As you can see, it is a special amino acid in the fact that there is only H there. So therefore, it has the smallest, and it also helps in offering very flexible structures because it only has the H there. We have other special groups such as cysteine. So cysteine is one of the special groups in the fact that it has got a sulfur on the side chain, which is able to combine to form a disulfur bonds. We also have other special things such as proline. So proline is able to form structures that are going to look like CH2, our CH2, then we have our CH2. It is able to combine with the nitrogen that was originally there like that. So this is our proline. So these are just a special amino acids. Other amino acids, on the other hand, have got what we call an aromatic ring. So though they might fall in the groups of polar, like for example, we might have a phenyl, phenylalanine. So phenylalanine has got the special properties in the fact that it has an aromatic ring. So it looks like something like this. So where we can have C, so same thing here, H, N, H, so HH there. Then we are going to have our CH2, then we are going to have this ring. So this ring is the characteristic property of those that have aromatic rings. We might have phenylalanine, we might have histidine, we might have tryptophan, we might have a lot of these. So amino acids can be either, we might have essential amino acids, and non-essential. So essential amino acids are those that the body cannot produce. Body cannot produce. So you have to get it from food sources. So the essential amino acids that we have, we have a total of 9, but some of the examples that I'll give you are on the essential amino acids are the phenylalanine, So we have the phenylalanine. We might have other examples such as we might have tryptophan. Remember, these have got at least know some. So we have tryptophan. These have got aromatic rings. We might have methionine. We have a lot of them. We have the valine. We have lysine. So there are a lot of them. Now, on the other hand, so these are needed mostly for protein synthesis and repair. So we have synthesis of proteins and repair. Now, when we look at the non-essential, so non-essential does not necessarily mean to say they are not important. No, non-essential amino acids are those amino acids which the body can produce. So if the body can produce, then that simply means to say they are non-essential amino acids. So we can have examples such as our alanine. We might have examples such as asparagine, glutamic acid, aspartic acid, serine. So even you can just remember the acids and all that. So these are the non-essential amino acids. Thank you very much for watching. Hope you would like, subscribe, and share for more. In the next video, we are going to be looking at levels of organization. See you in the next tutorial.